we wonder what will be the effect of the calcium activity on the neuronal network activity. And for that, we did the similar experiments, but now we also included a recording electrode in the primary somatosensory cortex to record the local field potential of the cortex. And here I'm showing traces of the representative trace of the local field potential recording with the electrode located in the primary somatosensory cortex. And here is the color-coded power spectrum of this signal, electrical signal. Here, the calcium elevation on some of the astrocytes in that area. And you can observe that upon the sensory stimulation in the hindpole, with electrical stimulation in the hindpole, we observe this type of responses, which is characterized by an enhancement of the gamma activity, an initial enhancement of the gamma activity of the field recording, gamma activity between 40 and 60 hertz, and then upon the continuous stimulation of that, they could produce a decrease in the gamma activity of the signal. This is a well-known phenomenon in brain physiology, which is called sensory adaptation. Upon the continuous stimulus, the neural network produces this sensory adaptation that the responses is decreased upon this continuous stimulation. You can see here that with this stimulation, with certain delay, there is a calcium elevation in the astrocytes we were recording from. So there was some kind of delay into these calcium responses of the astrocytes. If we plot here the time, here is the stimulation, and we plot the relative gamma activity and the astrocyte calcium signal, we can observe that upon the sensory stimulation, we have an initial surge in the gamma activity of the neurons that then reaches a maximum and then decreases to a steady state that persists as long as the sensory stimulation. This decrease in the gamma activity is what is known as the sensory adaptation. And then when we look at the calcium, we observe that the calcium elevation in the astrocytes on average is relatively delayed. And the rise in the calcium signal is temporally coincident with the decrease in the gamma activity. So when the calcium goes up, then at that time, temporally correlates with the sensory adaptation, the decrease in the gamma activity. We plot here one signal over the other, the astrocyte calcium versus the gamma activity, and we observe this, what we call the hysteresis loop of the astrocyte neural network interaction. So before, when we produce the stimulation, before the calcium in the astrocytes elevates, there is an increase in the gamma activity. And then when the calcium signal starts to raise, there is this decrease in the gamma activity, which is the sensory adaptation. So we believe that, or we hypothesize that this sensory adaptation would be due to the calcium elevation in the astrocytes that we were producing this sensory adaptation. To test that hypothesis, we use the IP3 receptor 2 knockout mice, in which the calcium signal in the astrocytes is largely prevented. So there is no calcium or not significant calcium elevation in the astrocytes in these mice. And we compare the effect of this continuous sensory stimulation and the sensory adaptation in these mice compared to wild type. So we observe here in the wild type mice, we observe this increase in the gamma activity, and then the decrease as we have seen the sensory adaptation. When we compare that with the IP3 receptor 2 knockout mice, we observe that now the sensory adaptation is much reduced. It's not as strong as in the wild type mice. We quantify here the gamma activity over time. We see here in wild type mice, we have this maximum reach of the gamma activity, then decreases to the steady state with the sensory adaptation. But we see here with the IP3 receptor 2 knockout mice, we have also this increase in the gamma activity and then the reduction. But if we compare the peak versus the steady state of the gamma activity, which reflects the sensory adaptation, we can observe that in the hyperreceptor 2 knockout mice, this sensory adaptation is much reduced, suggesting that the astrocytes are contributing to the regulation of the neural network involved in the sensory adaptation. To further test this hypothesis, we performed the following experiments to activate the astrocytes using the pharmacogenetic approach of expressing the GQ DREAD, specifically the somatosensory cortex, in the primary somatosensory cortex. And we recorded the gamma activity induced by the sensory stimulation in the absence or in the presence of the CNO, the ligand that activates these GQ DREADs. And we could observe that the responsiveness of the neural network, the gamma activity, is differently regulated in the presence or absence of the activation of the astrocytes with CNO. And what you can observe here, that the activation of the astrocytes reduce the responsiveness of the neural network to the sensory stimulation. So the activation of the astrocytes reduced the gamma activity induced by the sensory stimulation. They are kind of slowing down the neural network activity. The activation of the astrocytes are slowing down the activity of the neural network evoked by the sensory stimulation. Now we wonder what would be the effect of CNO in the basal conditions without sensory stimulation. What would be the effect of increasing calcium in the cortical astrocytes without sensory stimulation? We performed the same experimental approach of expressing the GQ DREADs in astrocytes in the primary sensory cortex, and then inject CNO to activate those astrocytes. And what we observe is that in that cases, in the low range, not in the gamma activity, but now in the delta range, in the low frequency activity of the neural network, there is an enhancement of the activity of the neural network by the stimulation of the astrocytes. This idea, we propose the following hypothesis or mechanistic idea about the role of astrocytes in the neural network function. First, these data suggest that the cortical astrocytes respond to sensory inputs, that the cortical astrocytes regulate sensory evoked neural network activity, and that the cortical astrocytes maximize the functional dynamic range of the neural network. So when the network is very active during the sensory stimulation in the process of the sensory adaptation, they slow down the activity of the network by producing or contributing to the production of this sensory adaptation. However, when the network is in the resting state in the basal and we activate the astrocytes, we then produce an enhancement of the activity of the delta activity of the network. So we propose this homeostatic mechanism of the role of astrocytes in the cortical network function is that if the network activity is very high, for instance, during sensory evoked sensory stimulation, the astrocytes reduce the network activity, they reduce the gamma activity to move it to the left. However, when the sensory input is absent, or there is no sensory input in resting states, the astrocyte would enhance the delta activity to move also the network activity to this medial point here. The reason for that, or the advantage of that, the biological advantage of that, is that when you have the network activity in the middle point, then you can have a huge increase the functional dynamic range. Small sensory input changes could produce a huge changes in the network activity. If you are in this position when the network activity is in the very high activity, then changes in the sensory input would not produce major changes in the network activity. And the same here, if you are in the very low phase of the sensory input, then the network activity will not produce major changes. However, going to this medial point with small changes in the synaptic input would produce huge changes in the network activity, allowing then the network activity to be able to codify more efficiently the sensory synaptic inputs. how these changes in brain pathology, we were wondering what would be the effect of some brain pathology on this neural network interaction, astroglial neural network interaction. And for that, we have been studying the animal mouse model of Alzheimer's disease, which is the APPPS1, is a classical model for Alzheimer's disease. And we crossbred these animals with the GCAM7 to monitor calcium activity in the astrocytes simultaneously. And we also could monitor the beta amyloid plaques using this in vivo labeling with methoxy X04. So we could monitor the calcium activity in the astrocytes using this approach that I have shown before. We monitor the beta amyloid plaques, and then we monitor the calcium activity using the two-photon microscopy and the GCAM7 calcium sensor, and then we stimulated the calcium, the hindpaw, to produce the sensory stimulation. And we then compare in control animals. We produce the sensory stimulation and record the calcium activity in the arborizations and in the somas of the astrocytes. And the first thing we notice is that upon sensory stimulation, now the number of calcium events that we could detect were enhanced. Wait a second. The spontaneous calcium events that we were detecting in the astrocytes, in the arborizations of the somas, were enhanced in this APPPS1 Alzheimer's disease animal model. So the activity of the astrocytes spontaneously is much higher in these Alzheimer's disease animals. We also observed that the calcium signal in the astrocytes upon evoked by sensory stimulation was altered. And they have a much lower dynamics of the calcium events in these APPPS1 animals. So the astrocytes are more active, and the responses to this to the sensory stimulation was slow. But most interestingly, what we found is that the spontaneous activity showed a positive correlation with the beta amyloid plaque density. The more beta amyloid plaque density, the higher the spontaneous activity of the astrocytes. However, we observe a negative correlation of the responsiveness to the sensory stimulus in this, as in relation with the beta amyloid plaque density. That means that the beta amyloid plaques produce an enhancement of the spontaneous activity of the astrocytes. The astrocytes became hyperactive, but at the same time, they are hyper-responsiveness. They are less responsiveness to the sensory evoked. They are much more independent of the sensory input. They are hyperactive and hyper-responsiveness. Then we wonder what will be the effect on the neural network regulation. So we produced a similar experiment that we have shown before in control animals, but now in APPPS1, and we compare these, just to summarize the results in this hysteresis loop. And you can observe in the wild type, we observe this classical pattern of behavior of the gamma activity versus the calcium activity, with the initial gamma activity raise and then the decrease to steady state level with the sensory adaptation. And you can observe here how disrupted is this pattern in the APPPS1. We have a huge increase in the gamma activity, the reduction, and then some less adaptation and completely disrupted. So this hysteresis loop that reflects the astrocyte neuronal network, astrocyte neuroglial network interaction, is disrupted in these animal models of Alzheimer's disease. And we propose that maybe the cognitive impairment in Alzheimer's disease may be related to this dysfunction in the astrocyte network, astrocyte neural network communication. Now, another type of the work that we're doing in the lab is to test what is the effect of this synaptic regulation by astrocytes on animal behavior. We wonder whether this what we're doing is actually impact the animal behavior. And for that, we studied and we are continuing studying the role of astrocytes in synaptic transmission in the amygdala. Why the amygdala? The amygdala is a very complex structure, which includes 13 different subnuclei. So structurally, it's very, very complicated, but it has a great advantage regarding the behavior. They have a very easy readout behavior, which is typical fear of the animal that can be very easily measured and quantified. So the complex structure of the amygdala, however, we reduce it to something very simple and very schematic. So these different subnuclei, all of them funnel the information to this subnucleus, which is called the centromedial amygdala. All the information is related to the centromedial amygdala, which is the output of the amygdala. This subnucleus, the centromedial, is the output of the amygdala. And these neurons in this nucleus receive inhibitory input from the centrolateral amygdala and excitatory inputs from the basolateral amygdala. And all this information that is received in the amygdala then is funneled to this centromedial nucleus that produce the effector output of the amygdala. We could record in brain slices the excitatory transmission from the basolateral amygdala, stimulating the basolateral amygdala and recording the neurons in the centromedial nucleus. And we could observe, or we could record also the inhibitory synaptic transmission from the centrolateral nucleus and record it from neurons in the centromedial amygdala. And then we wonder what would be the effect of activating astrocytes into the synaptic transmission in these excitatory and inhibitory terminals in the centromedial nucleus. For that, we use this strategy of the DREAD strategy, the pharmacogenetic strategy of injecting the amygdala with this virus that contains the GQ DREAD and the GFAP promoter to express specifically the DREADs in astrocytes in the centromedial nucleus of the amygdala. We observed that upon activating these astrocytes with CNO, we could observe these calcium elevations in the astrocytes as expected. Then what is the effect of this calcium elevation in the astrocytes on the synaptic transmission in these neurons? So we recorded the centromedial nucleus and we stimulated the inhibitory terminals from the centrolateral nucleus. And we observed that before the activation of the astrocytes, we observe these synaptic responses, and upon activation of the CNO, we observe that these inhibitory currents were the probability of having these inhibitory currents were much enhanced, as quantified here. So the activation of the astrocytes enhanced the inhibitory synaptic transmission in the centromedial nucleus, and this was mediated by the release of ATP that converted adenosine that is blocked by blocking the A2A receptors. In contrast, when we stimulated the excitatory terminals from the basolateral amygdala and recorded the synaptic currents in the centromedial nucleus and now activated the astrocytes, we observe a decrease in the probability of release of the excitatory synaptic transmission. So the astrocytes enhance the inhibitory synaptic transmission in this nucleus and at the same time decrease the excitatory synaptic transmission in the nucleus. So they produce the enhancement of the inhibition and the decrease of the excitation. And based on the pharmacological profile, we propose that astrocytes activated by CNO produce the release of ATP that after being converted to adenosine, the adenosine would activate A2A receptors in the inhibitory terminals to enhance the probability of transmitted release. And at the same time, this adenosine would activate A1 receptors in excitatory terminals to produce depression of the excitation. In general, what they produce, the general outcome would be an enhancement of the inhibition of these neurons and depression of the excitations. The neurons would be much more silent. To test this hypothesis, we recorded in vivo the electrical activity of the neurons in the centromedial nucleus with these local field recordings, and you can see here these individual spikes in the reflecting the neuronal activity before and after activating the assays with CNO. And you can observe that the firing rate of the neurons, the spontaneous firing rate of the neurons, was largely decreased by activation of the assays, which is consistent with the enhancement of the inhibition and the depression of the excitation. And then we tested what would be the effect of activating assays in the behavioral response, knowing that this nucleus is the output nucleus of the amygdala. And for that, we used the classical fear conditioning paradigm, in which the animals were trained to associate the food shock with a tone. So we deliver the first day a tone and a food shock, and the animal, as a response, as a fear response, they freeze, and we quantify that freezing. The first day, we observe the freezing with a tone, the conditional stimulus, and then conditional stimulus with the food shock, and we observe that the animal learns that after the tone, there's a food shock, and they don't like it, and they freeze. Now, the following day, we then produced the test of the fear expression, just delivering only the tone, the conditional stimulus, to see what is the behavior of the animal. We observe in normal animals or in control animals, they have learned that the tone produces the food shock and they freeze. However, in some animals, we injected previously CNO to activate the astrocytes, which we know that decreased the firing rate of the neurons in the centromedial nucleus, which is the output neurons. And we observed that the animals now, the freezing behavior of the animal is much reduced, which is consistent with the fact that the centromedial nucleus neurons are silenced by this enhancement of the inhibition and the depression of the excitation. Then, as a conclusion of the work I have presented, that the associated regulation of serotonin emission is a subtle and complex phenomenon that occurs at the specific synapses in specific circuits. It's not a general phenomenon. We have seen here, and one of the examples I'm showing is this in the amygdala, with this, the assays could regulate in one way or the other, either the excitation or the inhibition. So they can produce this complex signaling that specifically signal in specific synapses differently. I have shown also that assays sense and regulate neural network function, that the sensory evoked assay signaling and assay neural network interactions are altered in an Alzheimer's disease mouse model, that maybe we propose that maybe the dysfunction of these assay neural interactions may contribute to brain pathophysiology. So like the cognitive deficits associated with Alzheimer's disease maybe are due or are partially contributed with this dysfunction of the assay neural interactions. And finally, assays influence animal behavior through the selective regulation of specific synapses, as we have seen in the amygdala. And I want to finish with the acknowledgements, the current people in the lab, the previous members in the lab, the collaborators, and of course, the funding institutions that they make this work possible. And thank you very much for your kind attention. And if you have any questions, I will be happy to answer.
So thank you, Alfonso, for a great talk. So let's see if from the auditory board there is some question. So not yet.
Sebastián has the hand raised.
Ah, sorry, Sebastián, I was not looking to you.
Yes.
Please. No.